Alrighty, this is right where we left off with Chapter 7. We're going to start going over the brain. We've gone through the structures of neurons, the neuroglial cells, as well as action potentials, and some protective measures such as cerebral spinal fluid and meninges. To start with the brain, we'll start with the outermost. I call it the brainy part of the brain. It's called the cerebrum. It's divided into lobes, the frontal, parietal, temporal, and occipital lobe. These lobes all correspond with the bones, which they're underneath of. You can see here this diagram goes through some of the special areas within each lobe. We'll start with the frontal lobe, which is over here. So you can see that this area over here would be the front of the body, or the ventral side. Back here is posterior. So this would be the part that's just above our eyes. Our frontal lobe is sending motor output. It contains this special area highlighted in pinky red. This is called the primary motor area. It's on what's called the precentral gyrus. The gyrus is, or gyri, is plural. That's a technical term for these mounds that are on the brain. The grooves are called sulci. The mounds are called gyri. So this particular mound is called the precentral gyrus, and this is where our primary motor cortex is found. So all of your motor coordination, movements, dancing, etc. is all done through this specific area of the brain. There's also an area called the premotor area. This is where some coordination comes into play. Broca's area is a speech center. If you think of speech as a motor movement, that is very true. Think about your tongue and lips and your vocal cords, how everything has to flex and move in order to make those sounds. So that's within that primary motor area. You have a frontal eye field area located on your frontal lobe, and the prefrontal lobe, which is in front of all of these structures. This is where abstract thinking is, decision making, judgment, intuition, learning, memorization. All of that is all in this area of your brain. So y'all want to work to develop that part of your brain, right? If we go to the sides of our head, we get to our parietal lobe. Our parietal lobe is our main somatosensory information area. This blue gyrus is the post-central gyrus. It's called post-central because it's just after this central sulcus, which divides the brain into anterior and posterior parts. So we had our pre-central gyrus of our motor, and now we have our post-central gyrus with our primary sensory. We also have a gustatory area or taste area of our brain, as well as a somatic association area. There's also an integrative center where some major integration of various senses come into play. We go over to our occipital lobe, or I should say back to our occipital lobe. This is our visual cortex. This is where all of our visual information is perceived. Our temporal lobe has a special area for interpreting speech called the Wernicke's area, as well as the auditory association area. This is where all of your hearing interpretation takes place. Whoops, sorry, my mouse went crazy again. The two halves of our brain are joined by a structure called the corpus callosum. You can see it here in this structure. Corpus callosum, basically a bunch of projection fibers going from side to side. This allows information from the right side of the brain to communicate with the left and vice versa. Otherwise, the two halves of our brain, while pretty much symmetrical, are separate. There's also structures called the basal ganglia. Here you can see them highlighted in blue. They're special areas that control background movements, such as walking. Once you learn how to walk when you're an infant, and not when you're an infant, but when you're a toddler, once you learn how to walk, walking then becomes one of those ingrained, almost reflex-like behaviors, and you can do other things while you walk, such as chew gum and talk on the phone and think about what you're going to wear the next day, etc., etc. So that background pattern of movement is controlled by your basal ganglia. Here you can also see some special structures that you will go over in labs, such as that longitudinal fissure dividing the brain into right and left hemispheres, structure called the thalamus, which we'll talk about in a little bit. The internal capsule is kind of like the meat of the brain. And then some other structures here, such as the hypothalamus, optic tract, and third ventricle. We'll get into all of these soon enough. So let's talk about the other parts of our brain other than the cerebrum. We have the brainstem, the diencephalon, and the cerebellum. So we'll start with the topmost structure called the diencephalon. The diencephalon, if you were to remove the cerebrum, the diencephalon almost looks like a lollipop structure inside of the brain. It is composed of really four separate structures. One would be the thalamus. The thalamus is this kind of dark blue area. I know my colors. Dark brown area. Oh my goodness, it's very late, guys. Sorry. So you have this dark brown area called the thalamus. This is where all of our sensory information is relayed. So you have sensory information coming in via afferent fibers coming up the spinal cord. It's going to go to the thalamus, and the thalamus will say, oh, okay, we need to send that information up to our primary sensory cortex right here. So that would be the job of the thalamus. The hypothalamus is just underneath and in front of the thalamus. It's this area right here. The hypothalamus is the relay between the endocrine and nervous systems. So the hypothalamus is going to control a lot of our autonomic nervous system functions, the pituitary gland, emotional states, eating, drinking, sleep-wake cycles, etc. The pineal gland is another structure of our diencephalon. It's on the other side of our thalamus, and the pineal gland secretes melatonin, which is important for our sleep-wake cycles. When it gets dark outside, our body starts to secrete melatonin, which is why we get tired at night. The next structure that we'll talk about on the other parts of the brain is the cerebellum. This is this other part of the brain. It almost looks like a nut coming off of the back of the brain. This structure is attached to the brainstem, and it is where all of our body position and coordination as well as posture is regulated. So individuals that do gymnastics and such, they work their cerebellum a lot. Things like organisms like cats and squirrels have very large cerebellums because they are always balancing. Your brainstem is this literal stem region of your brain, kind of supporting your brain on top of your spinal cord. It consists of three main areas. One's called the midbrain. The midbrain's right here underneath of the thalamus and hypothalamus. Then you have the pons, which is this kind of circular area here. And then the medulla oblongata, which is the last stop on the way out of the brain. So the midbrain is in charge of a lot of basic reflexes that deal with the head, eyes, ears, etc. If you see a bright flash or hear a loud noise, the reflex that makes your head turn to look for it is all coordinated through the midbrain. The midbrain is also called the mesencephalon. The pons contain sensory and motor tracts that help regulate breathing and respiration. The medulla oblongata also controls rate of breathing as well as heart rate, blood vessel diameter, and blood pressure. Some autonomic, actually most autonomic functions are also relayed through the medulla oblongata. The homunculi are groups of neurons that are devoted or traced back to specific sensory or motor areas on the cerebrum. A homunculus is basically a map within the sensory and motor areas showing the amounts of cells dedicated to that given area of the body. So here you see the sensory and motor homunculi. In this case, we're looking at the somatosensory area, and you can see that this area of the body is dedicated, or brain rather, these neurons are dedicated to the foot. This area is dedicated to the genitalia, so you have a very small number of neurons dedicated to that. Whereas if you look, for instance, at your hand, you have lots of sensory information coming in from your hands, lots of sensory information coming in from your lips, and you know that because this area, this picture of the face is very, very big. That's denoting the number of neurons dedicated to that particular area of the body. Likewise here, you have the homunculi for the motor neurons. So you have many, many motor neurons dedicated to your hand, many, many motor neurons dedicated to your mouth and throat for vocalization, salivation, etc. So the homunculi, as I mentioned, where the maps appear to be distorted, they're reflecting areas that have either more or less neurons associated with that body part. Areas where fine motor control, like the hand, have greater sensitivity. Areas where the mouth is where more of the cerebrum is dedicated to that particular function. In areas with fine motor control, the motor unit is going to be smaller with more numerous units for greater sensitivity. Places requiring more gross motor movements have large motor units and less overall neurons associated with them. There is something called brain lateralization or brain dominance. You may have heard of your right-brained or maybe your left-brained. And that's because certain sides of our brain, right and left, are more sensitive to particular aptitudes. The left hemisphere has more to do with logic and linear reasoning. Math, as well as writing, is mostly dealt with on the left side of the brain. The right side of the brain is more artistic and musical, imaginative, as well as intuitive. So some people will say that they're more right-brained because they're more artistic. Some people will say that they're more left-brained because they're more math-oriented. The majority of the population is right-handed. The rest are left-handed. This makes sense. The majority of the population is actually left-brained. The left side of your brain controls the right side of your body, and the right side of your brain controls the left side of your body. Most artists are left-handed, and that again goes back to the right hemisphere dominating that artistic side of your personality. Let's move from brain and talk about the spinal cord. The spinal cord, of course, is found in that vertebral canal that going through the vertebral foramen, if you recall from the lecture on bone anatomy. It's about 42 to 45 centimeters long, about 18 inches or so long. It's continuous with the medulla oblongata. It's shorter than the vertebral column. The lumbar, sacral, and coccygeal nerves actually exit at different levels than where they exit the vertebral column. The cervical and lumbar areas have special enlargements that control upper and lower limbs. And they're also covered with meninges and surrounded by CSF, just like the brain. Here you can see the picture. You can see the cervical enlargement and the lumbar enlargement. If you were to get injured at the lumbar enlargement, you would probably be paraplegic. You'd be paralyzed from the waist down. If you were injured in your cervical enlargement, then you would be quadriplegic and paralyzed from the neck down. Each of our, sorry about that. My touchpad gets a little excited. If you look at this picture here, you can see the anatomy of our spinal cord with our vertebrae removed. You can see that the spinal cord has blood vessels running up and down it. You can also see the three separate meninges, pia, arachnoid, and dura mater, which would be the most superior. You can see these yellow fibers coming in or leaving the spinal cord. The one that has the bulb, and we'll go look at this image now, the side that has the bulb on it is the sensory side of our spinal cord. Sensory information comes in, and the cell body of that sensory neuron is within that ganglia, which is on either side of our spinal cord. That's on the dorsal side of our spinal cord, or the posterior side, and that ganglia is called the dorsal root ganglion, and every single spinal nerve has a dorsal root ganglion associated with it. The ventral side, or anterior side of the spinal nerve, is the motor side. I remember that because I'm always moving forward with my life, and the motor neurons are on the front or anterior side of the spinal cord. You can also see some of the other structures in this image. You can see that central canal in the very center. This butterfly or H shape is the gray matter. Gray matter is made of unmyelinated axons as well as cell bodies. The white matter, in this case, surrounding the gray matter, is made of myelinated axons. So you can almost imagine tracks of axons coming into and out of this PowerPoint slide, which is carrying information up to the brain or down from the brain in ascending and descending tracks. So as I mentioned, the spinal cord nerves enter and exit from each segment of the spinal cord. The cell bodies lie outside the spinal cord, of those sensory neurons rather, in that dorsal root ganglion, as I mentioned, and the cell bodies of the motor neurons lay within that central area, that central gray area of the spinal cord, that gray matter. The white matter that surrounds the gray matter contains the myelinated nerve fibers that are divided into three tracts. There's an anterior white column, a posterior white column, and a lateral white column. I'll show you what that means. So if you look back at this picture, remember this is called the dorsal root ganglion because it's on the back side of the spinal cord. So this would be the posterior white column, this would be the lateral white column, and this would be the anterior white column. This would be the other lateral white column. So, shape and proportion of gray matter and white matter is going to vary with each spinal cord segment because there's a number of different functions for every spinal cord segment. Ultimately, the spinal cord is the relay and processing center for our peripheral nerves. The peripheral sensory nerves are bringing the information in. Before it gets to the brain, it has to go to the spinal cord. So the spinal cord is going to relay that information. There's nerve fibers from sensory receptor neurons at various levels of the spinal cord according to their receptor location. So in other words, the sensory receptor neurons from your legs are not going to enter your spinal cord at the cervical level. They're going to come in at that lumbar level. So here you can see what I mean with ascending and descending tracts. I mentioned that earlier. We look at the ascending or sensory tracts. It's called ascending because the information is going up or ascending in our body, coming in so that dorsal root ganglion on the back side of our spinal cord, coming up that posterior column pathway in this case, up through the medulla oblongata, which is going to be our major relay center for lots of our information. It then synapses with a second neuron at the medulla oblongata, and because we're talking sensory information, that sensory information goes straight to the thalamus. The thalamus is our major sensory relay center. It's going to synapse with a third motor neuron, which is going to take that information up to our primary somatosensory cortex within the parietal lobe, and then we can say, we just touched a hot stove, or oh, we just saw a bright light, right? That's the ascending sensory pathway. So our descending motor pathway is now going to carry information out of our brain. That upper motor neuron is going to carry information down through our medulla oblongata, which is our main relay center. That motor track is going to cross over, cross sides, if you remember. Right side controls left side of the body, left side controls right side of the body. You can even see that lateralization here. This side's going to the other side of the body. And then we're going to synapse with a lower motor neuron within that gray matter of our spinal cord, carrying out that ventral root of our spinal cord, and then out to the spinal nerve to whatever skeletal muscle that we need to control. Let's talk about the autonomic nervous system. I mentioned earlier the sympathetic and parasympathetic division. Wasn't sure if I had those slides on here, but they were just later. The autonomic nervous system, as I like to say, is the automatic nervous system. This is the self-regulating system. It conducts nerve impulses involuntarily that go to smooth muscle, cardiac muscle, as well as glands. It can be sympathetic or parasympathetic. Two-neuron pathway that connects our central nervous system to peripheral effectors. So in other words, you saw in the ascending and descending tracts, in some cases, we're looking at a 3-link or maybe a 2-link pathway into or out of the central nervous system. The autonomic nervous system is just 2 neurons. There's a preganglionic fiber and a postganglionic fiber. The cell bodies for the second motor neuron or postganglionic fiber are always within the ganglia. The ganglia, if you recall, are gatherings or clusterings of cell bodies outside of the spinal cord. They're usually within the wall of the organ, or they could be in chains that are on the side of the vertebral column or near large abdominal arteries, such as our aorta. So here you see a comparison of somatic and autonomic nervous systems. In this case, we had been talking about the somatic nervous system controlling through skeletal muscle. We have that motor neuron coming out of that gray matter, leading through that ventral root, traveling down a myelinated axon to whatever skeletal muscle needs to be stimulated. In the case of our autonomic nervous system, we have a different type of motor neuron here called a preganglionic neuron, leaving our gray matter. Again, coming out that ventral root because it's a motor neuron. It's then going to synapse outside of the central nervous system within a ganglion or a cluster of cell bodies. That postganglionic fiber is then going to carry out the command to whatever tissue. In this case, maybe the heart, maybe the pancreas, maybe the bladder. It could also, this example is a sympathetic example where we're using norepinephrine as a neurotransmitter. Sometimes our sympathetic nervous system innervates our adrenal gland. In this case, our adrenal gland is going to act as the ganglion. So we have our preganglionic fiber going all the way down out our spinal cord, all the way down to our adrenal glands, which, if you recall, sit on top of our kidneys. It's a very long nerve cell. And then within the nerve cell, it's going to synapse with other postganglionic fibers, which tell special cells within our adrenal gland to release epinephrine and norepinephrine, which can be distributed throughout the body via the blood. This is going to cause an excitatory response in lots of our muscles. Our muscles are going to get ready to react. Our heart rate's going to increase. Our digestive secretions slow down. Our respiration rate speeds up. Overall cell metabolism speeds up as well. In the parasympathetic division, again, we're synapsing outside of our central nervous system. You have that preganglionic motor fiber leaving through that ventral root of the spinal cord, traveling down to an autonomic ganglia. In this case, it could be a location near the heart or near the pancreas or near the bladder. But either way, that postganglionic fiber is going to carry the information directly to that organ. It's going to tell that organ either to slow down or speed up. The parasympathetic division tends to slow down our muscles, slow down our hearts, slow down our respiration, but speed up our smooth muscle contractions. In other words, speed up digestion. If you recall, parasympathetic is that rest and digest. So, let's get a little more detailed here. As I mentioned, the sympathetic division of the autonomic nervous system is that fight or flight response. It's also known as the thoracolumnar division because the sympathetic nerves are coming from the thoracic and lumbar segments of our spinal cord. The ganglia are on either side of the vertebral column or near the abdominal aorta or within the adrenal glands, which is not written on this slide. The neurons are using two different neurotransmitters. They could use acetylcholine. They could also use norepinephrine. Preganglionic fibers are going to release acetylcholine, and then postganglionic fibers will release the norepinephrine. That norepinephrine is going to have a longer lasting effect, which is why sometimes after you were very excited about something or nervous about something, you just can't shake that feeling of being nervous or excited. That's because the norepinephrine is remaining in your blood. So, a sympathetic division is going to respond to those E's: exercise, emergency, excitement, embarrassment. This is what I've been saying of increased heart rate, increased force of contraction, increased blood pressure, getting those blood vessels to dilate, which is called vasodilation, getting blood flow to the organs, getting blood flow to the muscles. Dilation of airways, increased rate of breathing, increased rate of glycogen breaking down, increased rate of fatty acid metabolism, releasing glucose, getting ready for that aerobic metabolism to make lots of ATP so you have energy to do things. It's also going to inhibit digestion and conserve the blood for muscle action. It's not that important that you just ate a Snickers and need to digest it. What is important is running away from the lion or whatever it is. So the sympathetic division is that fight or flight. It's not about digestion. It's about getting away and survival. Here you can see some of the effectors and neural pathways that would be used in the somatic nervous system as opposed to the autonomic nervous system. So before we were talking about skeletal muscles, that's mostly voluntary. In the autonomic nervous system, we're talking about involuntary responses using acetylcholine from the presynaptic or preganglionic fibers and norepinephrine from the postganglionic fibers, carrying excitatory or inhibitory information to various peripheral effectors. Here you can see that sympathetic division, as I said, sometimes called the thoracolumnar because it's coming from the thoracic and lumbar segments of the spinal cord. You can see how it goes all over to various parts of the body. You can see how it's arranged in this chain on either side. The ganglia, as I should say, are arranged in this chain on either side of our spinal cord, coming out to the pulmonary or lungs, coming out to our heart, going to our salivary glands, our eyes, etc. The parasympathetic division is the rest and digest. This is also called the craniosacral because these peripheral nerves come from our cranial nerves as well as sacral segments of the spinal cord. The cell bodies lie in special ganglia which are close to the target organ, so that they're not within chains next to the spinal cord or within the adrenal glands. They're literally next to whatever organ they innervate, so they're all over the body. The parasympathetic preganglionic cells are much longer. They only synapse with 4 to 5 ganglionic neurons, so they're limited to a specific target organ. In some cases, they're just limited to a part of an organ. Parasympathetic postganglionic cells are much shorter. The pre- and postganglionic cells only use acetylcholine, not norepinephrine. Stimulation of the digestive tract is going to lead to secretion of gastric juices, pancreatic juices, as well as saliva to increase digestion. You're also going to get vasoconstriction, decreasing blood flow to our muscles, shunting all of that blood towards our digestive tract to get the digestive system moving. It's also going to relax the urinary bladder as well as anal muscles, leading to increased urination and defecation. So this is why sometimes after a long meal or a large meal, you have to go sit on the toilet for a little bit, because that entire parasympathetic division has stimulated your digestive system. Here you can see where those nerves are coming from. Again, the brain through those cranial nerves, or the sacral segments of our spinal cord coming out to some of our lower structures, including our intestines, our kidneys, for increased urination, as well as external genitalia and internal genitalia. Here, this chart goes through some conflicting actions or antagonistic actions of the sympathetic and parasympathetic divisions. Just to give you some examples, the sympathetic division would dilate the pupils, in other words, make our pupils nice and big to let lots of light in so you can be aware of your situation. Whereas the parasympathetic division is going to make your pupils smaller, because you're resting and digesting during parasympathetic stimulation, so you don't need to let that much light in. Let's talk about cranial nerves. We've talked about spinal nerves. As we said, spinal nerves come off the spinal cord and go out to various parts of our body, carrying sensory as well as motor commands. Our central nervous system is going to communicate to the peripheral nervous system and the outside environment. The cranial nerves carry motor, sensory, or mixed fibers. So some of our cranial nerves will only carry sensory information from whatever effectors or whatever receptors back to the central nervous system. Some of our cranial nerves are only carrying motor commands away from our central nervous system, and some are like a two-lane highway carrying sensory as well as motor information. There are 12 pairs of cranial nerves. Cranial nerves 1, 2, and 8 are sensory information only. Cranial nerves 3, 4, 6, 11, and 12 only carry motor information. Cranial nerves 5, 7, 9, and 10 are mixed nerves, carrying both sensory as well as motor information. This chart, or this diagram I should say, shows you where all of the cranial nerves arise from. Again, your lab instructor will let you know if you need to be able to identify these specific cranial nerves on a brain model. This chart over here goes through the specific functions of the cranial nerves and what they do. Let's talk about spinal nerves. As I mentioned, spinal nerves are all mixed. They carry sensory neurons as well as motor neurons or motor fibers. A plexus is a network of neurons formed from those emerging branches that come from the spinal cord. So here you can see a brachial plexus. This is going to all travel down the arm and innervate parts of our arm. Here's the lumbar plexus going down our leg. The sacral plexus also going down our leg within a different bundle. The cervical plexus is innervating parts of our head and neck. So different groups of spinal nerves are going to be associated with various plexuses and then be predictable in their behaviors. Here you can see some of the specific nerves. Again, it's up to your lab instructor if you are responsible for knowing any of these nerves. Just to point out, for the most part, the nerves are named after the types of or the specific bones that they're around. For instance, the ulnar nerve is in front of the ulna, the radial nerve is in front of the radius, the median nerve is in between, the axillary nerve is in the axillary area, and the axillary nerve actually branches off to form the radial and ulnar nerves. So that's something interesting as well. The femoral nerve over here branching off to form a couple other secondary nerves, the tibial nerve, fibular nerve, etc. So spinal nerves in the thoracic region, T2 to T11, are called the intercostal nerves because they're not forming a plexus. And if we look back here, you can see that they're the intercostal nerves, not forming a specific plexus. A dermatome is a special area of the skin that sensory neurons are receptive to. Nerve damage can be assessed by touching specific dermatomes and determining if stimulation is perceived. So they wouldn't have to necessarily do an MRI or a CT scan to determine nerve damage in that case. They could just touch specific areas of your skin, and if you don't feel it, then they know that that particular nerve is damaged. There's small overlapping of some dermatomes, so anesthesiologists may have to block several nerves to prevent pain perception in a particular area. This chart goes through specific plexuses as well as listing the specific nerves within those plexuses and what they innervate and what they either control or sense in terms of sensory input. Again, it's up to your lab instructor if you're responsible for these. I think for your lecture portion of the class, you should at least be familiar with the various plexuses and what the plexuses innervate, but I'm not going to test you on specific nerves. Let's talk about reflexes. Reflexes are fast responses to a change in an environmental stimulus, ultimately to restore homeostasis. Reflexes are formed by a pathway called a reflex arc. It involves a sensory receptor, which is receiving the stimulus, a sensory neuron, which is going to carry the impulse back to the central nervous system, some type of integrating center for information processing, then a motor neuron, which is going to carry the motor command back out, and then the effector, which is going to respond. Usually it's a skeletal muscle. Here you can see the diagram using the patellar stretch reflex or the patellar tendon reflex. The doctor or lab partner or whoever bangs against that patellar tendon, stretching it slightly. That stretch stimulation is noted by a special sense receptor called a muscle spindle. The muscle spindle is going to send its information via the sensory neuron back up through the dorsal root ganglion, through the back side of our spinal cord where all of our afferent information comes, and it's going to synapse with our motor command, our motor neuron here within the gray matter, and that motor command is going to come out via that ventral root of our spinal cord, go out to our quadriceps muscle, and say, okay, we need to flex our quadriceps muscle and kick this leg out, extend our leg. Spinal reflexes occur only in the spinal cord. So this example here would be a spinal cord reflex. Cranial reflexes, like our pupil dilation, contractions in response to changes in light intensity, those reflexes go straight to the brain. So not all reflexes are synapsed in the spinal cord like this is. Sometimes the brain has to do with our reflexes as well. And that concludes chapter 7.